Welcome to today's Wednesday seminar. So a few of you might know that I did my PhD at the Basel Institute for Immunology. The ultimate honor in Basel was that if there was a foreign speaker to be introduced, especially somebody who had worked there before, it was to give that person no introduction whatsoever. And the reason was, it means you're so famous, you don't need an introduction. So I will not give Marco an introduction. He has contributed a huge amount to this institute already, especially in the last 2 years, by developing new technologies that probably about 99% of the people in the room have already benefited from. So I will not take up any more time. So today's seminar's title is Editing the Genome in Cells and in Mice Using CRISPR-Cas9 Technology. So, Marco.
So, yeah. Can you hear me, yeah? Okay. Thanks, Andreas, for the nice introduction, or non-introduction. And as Andreas said already, I'm talking today about the CRISPR-Cas9 technology. And most of you will probably know that I'm working, or my main research interest is in apoptosis and cancer. But I have also a very strong interest in technology development, and that's where it comes from, though I developed this technology. And I just want to show you, share some data with you and what we have done and what is possible to do at the moment with this CRISPR-Cas9 technology at WEHI. Just as a start, the top doesn't show up, confidentiality note that this seminar includes some information that is of potential commercial value, and don't talk too much about it and take it outside. Okay, so let's start. So I wanted to start my talk today by showing you on how important the CRISPR-Cas9 technology has become over the past 2 years, and it was really an explosion in the biomedical research. And these are the two people actually who have pioneered this, and this is Jennifer Doudna and Emmanuel Charpentier. And as a result for this pioneering in CRISPR-Cas9 and that this has become so famous, they were named this year in the Times magazine as they were on the list of the top 100 most influential people in the world for the creators of genome editing. So that, I guess, is quite a huge contribution. At WEHI, we have also established a small team to implement the CRISPR-Cas9 technology. And also, we are not quite as stylish as these two ladies. I think Tony Handy would be very happy with us because we take eye safety quite serious. But in all seriousness, I have to say a lot of data I will show today was generated by these two absolute stars in my lab, and this is Lynn Tai and Andrew Koo. And I'm really very thankful to them as they have done a really tremendous job. So over with that, and let's start. So the outline of my talk, I have divided it into 3 different parts. So we start off with show you something how we established the CRISPR-Cas9 in an inducible lentiviral system. So it is useful for any cell type you're interested in. And in my lab, since we are interested in mainly hematopoietic cells, lymphoma, and so on, we needed a system where we could access these cells with. The second part will deal with the generation of mice using this CRISPR-Cas9 technology. And in the third part, the shortest part of this talk, will also show you some results and some outlook on how you can use CRISPR-Cas9 for whole genome screening in vitro and in vivo, and what is done here at WEHI. So CRISPR, so what is CRISPR? So most of you will know what CRISPR is. It was identified or found in the prokaryotes as an adaptive immune system. And it only requires, in principle, 2 components. And these 2 components is an endonuclease called Cas9 and a so-called single guide RNA. And as the name suggests, this guide RNA or small guide RNA guides Cas9 to a genomic locus. So the guide RNA itself is an RNA which has a specific sequence. And with that sequence, you can target it to any genomic locus within the cell. Once it is there together with Cas9 just basically introduces double DNA strand breaks, and these DNA double strand breaks puts the cell into an emergency state because it has to repair, tries to repair this as fast as possible, and it does this in a very, very bad manner, and this is a non-homologous end joining process. So that means it's not properly sealed anymore, but it has done this with some errors. And these errors, well, you get an insertion, deletion, bigger deletions, smaller deletions, or just a mutation. They will usually inactivate your gene either by mutating active sites or by causing a frame shift and thereby abrogating gene expression. So as I said, this system was described by many publications in 2013, coming out in Science, Nature, and so on, but these original publications all used conventional plasmids that transfected cells. So we were put back in the time where we started transfecting cells with non-viral vectors, and it is quite hard to get into cells such as hematopoietic cells. So we said we need another system, and since my background comes from lentiviral technology, I said, okay, let's start with that. Let's adapt the system onto a lentivirus. And we did it fairly simple. So we generated a dual lentiviral system. So the first part of that encodes the endonuclease Cas9, and we have linked it via the T2A peptide with the M-cherry protein. So that means you're capable of identifying cells which have been transduced with your Cas9 protein based on the M-cherry expression. And then we have a second part of the system, which is a guide RNA expression vector. And this expression vector has a GFP gene, so you can identify cells which have been transduced with cherry and GFP by fax, and so thereby allows you to sort these cells. And then you have the expression, or you have a cassette which allows the expression of the guide RNA. Now on top of this, what we did, we put this cassette, we basically introduced the TET repressor binding site. So thereby, a TET repressor, which is encoded on the same plasmid, binds into this H1 promoter and prevents the transcription of your guide RNA. Now, if you add the doxycycline to the system, this would release the TET repressor, and ideally your guide RNA is expressed, and only then you will induce your knockout. So, as I mentioned in the beginning, one of my major research interests is apoptosis, and apoptosis in cancer and in lymphocyte development and so on. And so just for those of you who are not familiar with that, so apoptosis, we are interested in the intrinsic apoptotic pathways mainly regulated by the BCL2 family. And upon a death stimulus, you induce the expression or activation of the so-called BH3-only proteins, which then their job is basically to activate Bax and Bak. And it can do that in two ways, either by inhibiting the pro-survival antagonists, by binding them, which the pro-survival antagonists say keep Bax and Bak in check, and thereby they will be released and Bax and Bak are activated, or they can directly activate Bax and Bak. And the consequence of Bax and Bak activation is oligomerization, and then at the mitochondria, you get the mitochondrial outer membrane permeabilization, cytochrome C release, caspase cascade, and so on, and the cell is killed. So these proteins involved, our division and my lab, they are very interested in these BH3-only proteins and pro-survival proteins, and one in particular, and Andreas is the one who has defined, together with Philippe, the role of the pro-apoptotic BIM gene. And we thought, well, since we have all the expertise and the reagents in the lab to look for BIM, and we're interested in BIM regulation in general, we thought, let's start to test our system in terms of knocking out BIM in several hematopoietic cell lines. And that was our first choice. And so we went ahead and looked to design guide RNA. So what is required for designing a guide RNA? It's actually not very much if you look for the Cas9. The guide RNA only requires basically an HEG, which is called a PAM site. This is a protospacer-associated site. So you look in the genome for an HEG, and everything, or an NGG, whereby N can be any base, and then everything 20 bases upstream will be your guide RNA. So you incorporate these 20 bases into the lentivector, and then ideally we have in this lentivector also the structural bit of the guide RNA, which will then be expressed, and then it would target this to the BIM gene, which is here in exon 2, which is a start exon of the BIM gene. The expectation would be, if it is targeted, Cas9 binds, cleaves, and the gene will be inactivated. So we tested this, and we looked into a myeloid cell line, which is the FDCP cell line. We introduced two different mouse BIM guide RNAs, one human as a control, and this is an empty control. And then prior to dox and after dox, and you can see the system is not completely leaky-free. You get already knockout in the beginning, but you can see quite clear that upon addition of doxycycline for 3 days, you get a clear reduction in almost absence of the BIM gene. Now, if you look in the long-exposed Western blot, you can see there's still remaining BIM protein, and you might ask, why do you have that? You're not saying it's a knockdown, but a knockout. Wouldn't you expect complete knockout in these cells? Well, in this case, we have not looked for single cell clones. We're infecting a whole population of 1 million cells, and probably we have targeted about 900,000 cells, so 10% are left, which express the normal BIM gene. So that indicated already that the efficiency was quite high, and compared to the original vectors, which were described, where they described about 5 to 10%, we are up here probably up to 80% efficiency in the knockout. Now then we went on because we had to prove that this is not an off-target or something else. And we had to, of course, sequence. And at that time, we didn't have a sequencing platform. So what we did, we basically generated first single cell clones from these populations. And here's an example. These are all single cell clones named A1 to A8. And you can see all of them, or most of them, have no BIM protein left as compared here in the wild-type control. And then we cloned the genomic region out where the guide RNA should bind and sequenced the individual clones. And you can see, indeed, this is a wild type. And then we found at that time we get a T insertion leading to a premature stop codon. And then you have insertion of 2 base pairs, which lead to frame shift and also inactivation of the gene. So next step was, well, we could use it in the mouse cell lines. Will it also be possible to use it in human cell lines? And so we went on and transduced the human Burkitt lymphoma cell line, which is called RAEL. And we generated single cell clones straight away. And you can see there is some clones still express BIM, while others have lost it almost completely. And then we wanted to know, of course, well, we have inactivated BIM based on Western blot results, but is BIM really functionally inactivated, or have we accumulated something else? And so what we did, we used a typical BIM stimulator, which is ionomycin, which will kill B cells, which Burkitt lymphomas are, upon the calcium influx into the cells in a BIM-dependent manner. So when we did this, and this is shown here, you can see these are control guide RNA transduced cells. They die very nicely. So what we see here is viability of these clones. You can see upon addition of ionomycin, all these control clones don't die. And these are our knockout clones, which you can see here. And here's, for example, clone E1, where you have some BIM remaining. And it's quite nice to see that they die halfway through. So it's probably a heterozygous BIM clone. Whereas the others are almost staying completely alive. For example, B1 clone, which is seen here. There's no BIM, and ionomycin treatment doesn't harm that clone anymore. So we could also functionally inactivate this gene. The next step again was we had to identify whether we really introduced the mutations. And the cloning procedure, as I described before, just PCR amplifying, putting in a vector, and so on, is quite laborious. And lucky enough, we have the next generation sequencing machinery and applications built up in the systems biology division. So we thought, well, let's swap to that system and do the next generation sequencing. So together with Liam and his people, we have established a protocol which allows you to basically now, with just ordering 2 of your primers for your gene of interest, to identify any intel which you generate. So what we did is we have designed it in that way that we have a guide RNA target site, and then we put a primer pair upstairs and upstream and downstream of this site. But these primers contain additionally a unique sequence. So what you generate is then a sequence with a unique sequence at the end. And for these sequences, we have in the systems biology up there, we have a palette of forward and reverse primers. It means when you put it on the next generation machinery, you need the linkers and you need the barcodes in order to identify which specific reaction you're working with. And so what that means, you have an indexed barcoded primer, which then can bind your PCR amplicon, and then this can be put on the next generation sequencer and sequenced by the MiSeq reaction. And that this works is shown in the next slide very nicely, because this is now the difference to what we saw before. Before, we only got a taste of what has happened in these cells. But now when you look in this, the top one is basically the wild type. You might not be able to read the letters, but what you see in red here is what is mutated basically all the way down. And here's one which is not mutated, and that's a clone which still had wild type potential and was still BIM sufficient. So this system works pretty efficient and pretty well, and is quite cheap. Now, I told you we have developed this lentiviral system because it's inducible, and up to now I just showed you that we knocked out BIM, so you might ask, why would you need an inducible system? Because if you take away BIM, cells will love it, anything which they harm. Why would you do that? Well, because we are also interested, as I mentioned, in the other side of the BCL2 family, in the pro-survival BCL2 family members. So obviously, these members are there to keep the cells alive. So if I take some of these pro-survivals out, cells will have a disadvantage, you will lose them, they will die, and so on. And one of these pro-survival members, which we are really interested in, is the pro-survival MCL1 protein. And we have gathered a lot of data, and the division in particular over the past 5 years, that MCL1 is pretty important in AML, in MYC lymphoma, and so on. And based on this work, we thought we want to see whether we can, is our system powerful enough to delete MCL1 and reach similar results as we have seen with genetic deletion of MCL1 before. So we looked in a cell line, which is, again, the RIAL BL cells. And these cells are highly dependent on MCL1. And this is just an example shown here. We have since recently an inhibitor for the MCL1 protein. And you can see when you treat these cells with this MCL1 inhibitor in different concentrations, this is viability, you see that these cells die pretty quickly. This is shown here after 24 hours. If you add QVD into this mix with the MCL1 inhibitor, you can see you prevent it. So that clearly indicates QVD is a caspase inhibitor. It's an apoptotic process, and it's specific for MCL1. So this inhibitor has been tested in several different scenarios. So it really targets MCL1. So these cells, what that means is that this RIAL Burkitt lymphoma cell line is highly dependent on their growth for the MCL1 protein. So what we did then was we designed guide RNAs for the MCL1, 2 independent ones, and wanted to see whether the expression of this guide would lead to a similar outcome. So we started off with that. We generated these cell lines, so this is a Cas9 only, and then we had 2 independent guide RNAs. And you can see in response to doxycycline, which is here indicated by plus, after 3 days, you get a certain amount of killing, but the killing efficiency is not really as dramatic as you saw it with the MCL1 inhibitor. Now we wondered this could be a problem of the whole population, and you have to go down and generate single cell populations, because you might have some which kill very well and others not. So we did this. We cloned these cells out prior to DOX addition, so that means MCL1 was always present. And then we basically treated these cells in the single cell clones. And you can see, indeed, the reason for this lower efficiency of dying was that you had different populations within the pool, because some of them, indicated by the blue arrow, would not respond to the MCL1 guide RNA, while as others would respond. That means they will be killed by the induction of the MCL1 guide RNA. Now, that is all fine, and we thought, oh, that works pretty well. But the question, of course, is, and to make it workable and useful in doing some research with these cell lines, will this, for example, clone which is shown here, which react the second time the same? Now, the expectation when we started this was not, because it's a random process which happens in the cell, and you would expect one time you got a frame shift, the other time the system wouldn't cause a frame shift. So what we did, we repeated this experiment, and we were quite surprised what we found, because the cells which responded the first time or didn't respond reacted the second time exactly the same. And we did it a third time and a fourth time and a fifth time, and it was always the same. They always reacted in the same way. Now then we summarize this whole experiment, and what you see is you have some non-responders and some which react pretty well. That means die well when you take away MCL1. And this very nicely correlates also with the insertion of indel mutations in the MCL1 locus, because here's a frequency shown of non-homologous end joining events in the MCL1 locus, and you see that the clone D8, which reacts the best, has the highest amount of insertions in the MCL1 locus over time. And then we wanted to see whether these cells really lose MCL1, so then we looked on the protein level. Obviously, we treated these cells, we pre-treated them with QVD to avoid them from being killed. And you can see that the clones D8, for example, again, is a best deleter, and you see almost the whole MCL1 protein is gone, while others still have a little bit left. So this worked quite well. It was surprising, but it did work well. So the next step we thought, of course, could we use these cells or responder and non-responder clones also in a more physiological environment? Can we use these cells and put them in vivo in a xenograft model? And that's what we did in the next experiment. Basically, we used some responder clones, 3 here, and some non-responder clones, which are 3 here, put them into the NSG mice and let them grow for 2 weeks. We super-infected these cell lines before with luciferase, so we can image the tumors while they were growing. So we did this, and then we gave some mice doxycycline food, which would induce a guide RNA for MCL1. And what you can see here is that 5 days after the food, or dox food, was given to these mice, in the responder clones, we got a reduction in the tumor cells, whereas in the non-responder clones, the tumor cells would grow out and grow further. So this indicated, indeed, that we can use this system even in a more physiological xenograft model. Now, coming back to the surprise that these clones always reacted the same way when we treated them, what was the reason for that? Well, we had a look at the nature of the indels, not on the frequency, but we just checked what is the nature. Is it always the same mutation you're getting? And so we did this only we did it for all clones, but this is a representative example for the strongest killer. And you can see, quite surprisingly, we always found that we got G deletion. In 3 replicates, you see replicate 1, we got 93% were G deleted, replicate 2 as well, and replicate 3 as well. So we got a very low, basically, variation in the indel formation, which was surprising at that time and hasn't been described. When we were sending this for publication, then some of the reviewers came back, actually 2 out of 3 came back, said, well, this phenomenon has been described before, and it could be the so-called microhomology-mediated end joining. So people were thinking up to now, and I think still that is happening for many genes, that usually CRISPR induces a non-homologous end joining. But there is a second process which has been overlooked by then, and this is a microhomology-mediated end joining. What that means is you induce a break, the 2 strands slide into each other until they meet some sequences which fit again, and then basically they join, and that's it. But what they also could do, they could also take some from the neighboring chromosome and so on, which is homologous and repair. And this is probably the explanation why we get in our MCL1 always a similar mutation, because the 2 strands use always the same matrix to repair the error which has occurred. And when we look at all and it's usually you only get a deletion in the microhomology-mediated end joining, and when we looked at all MCL1 clones, we only find deletions, we never have any insertions. So that is at least for MCL1, and for others we see it as well. It seems like a common thing used by the system. Okay, so now we went along and thought, well, so we've shown now we can use it in cell lines, but the next question was, can we use our system in primary cells as well? And so what we thought was, since we are interested in the hematopoietic system and we are used to infecting fetal livers with anything and reconstituting mice with manipulated fetal livers, could we use the system and manipulate the hematopoietic system in a way that we can knock out genes? So what we did was we isolated from E13.5 fetal livers, transduced these fetal livers with a Cas9 and some guide RNA, and then reconstituted mice and analyzed the phenotype. So the idea would be you get a knockout within the hematopoietic system, nowhere else, and it is a very fast way of generating knockout mice. So we chose a system which is the p53 tumor suppressor system because we are interested in that as well in the lab. And just for those of you who are not as familiar with that system, so p53 is induced, amongst many other things, in response to oncogenic stress, such as the abnormal expression of c-MYC or BRAF, and then p53 is activated and induces, simply said, apoptosis. There's many other things again happening, but simplified apoptosis. Now, Jerry has developed the nice EMI-MYC mouse model, lymphoma model, where it's shown if you basically let these mice, they express the MYC transgene under the control of the E-mu heavy enhancer, that means in the B cell lineage. And these mice also come to lymphoma development with a median latency of 100 days. Now, if you take away only one allele, and this was done by Eva in Andreas' lab, one allele of p53, you can see that basically after 40 days, all these mice, all of them succumb to the lymphoma. And this, of course, was an ideal system for us to test whether we could mimic the same scenario with using p53 guide RNAs. So we did this, and we expressed 2 independent guide RNAs targeting sequences in exon 4 and 5 of the p53, and we found, in fact, a similar result as was shown for the p53 knockout. So the tumor development or the lymphoma development in the EMI-MYC mouse model is accelerated to a similar extent as in the p53 knockout. So we wanted, of course, to check whether we also have lost indeed p53 gene and not only accumulated something else. And so what we did, we checked, we generated, because this mouse model is quite nice to generate also in vitro B lymphoma cell lines. We generate these cell lines, and then we checked what we had based on p53. And so there's something to say about p53. Usually it's not visible in cells upon normal conditions because it is degraded constantly by the proteasome by MDM2. This is the E3 ligase, which does that. But then if you have a mutation in p53, it inactivates it, then you can see p53 suddenly visible. So that means it's inactive p53. So what we saw now was that we got in our panel of lymphoma cells not only no p53, which doesn't tell you anything at this stage, but we got a few which are appearing, which means you have mutant p53. And this is quite interesting because what we did with that system was now that we randomly, under this selection pressure, generated a protein of p53, which has a mutation which inactivated this gene. You could imagine to use this for other purposes. If you have an enzyme, you want to have it hyperactive, it gives an advantage for the cell. You might be able to put a guide RNA on that and thereby get an active enzyme in your cell or whatever. So that's quite interesting and quite a, I think, good thing to work with. Now, of course, we want to make sure that the other ones are also knockout. And what we did was, in order to test this, you can induce p53 in cells, which have wild-type p53 if you basically destroy the DNA with a DNA-damaging agent, such as etoposide. And that's what we did. So you induce, you give the cells etoposide, and you see in a wild-type cell, p53 comes up. Now you look in our immune lymphoma lines, nothing has happened. So no p53 protein comes back. And then when we checked whether functionally p53 was inactivated, you can see the same. These are all the lymphoma lines which we generated, which had no p53 or mutant. They're all resistant to etoposide treatment, so they're not dying in response to DNA damage. While here are 3 control cell lines, they die, and here's a knockout p53 controlled, conventional knockout. This reacts the same as our guide RNA-generated knockouts. And then just tell that it's really p53. We sequenced again all the lymphomas, and they all have mutations which lead to frame shift and so on. When we summarize again, we find again similar occurring mutations within the p53 targeted regions. So that's so much to the in vitro system using CRISPR-Cas9. And now I wanted to switch gears and come to the second part, which will deal with the generation of mice using the CRISPR-Cas9. So the conventional way of generating knockout mice is, I don't know what that is now. What does it say? Well, you can imagine, yeah, good. So the conventional way of generating knockout mice, which was, the Nobel Prize was given to these people who invented this, which was Mario Capecci, Sir Martin Evans and Oliver Smith in 2007. It's a very important technology to manipulate the mouse genome and basically investigate the function of genes in vivo and so on. However, the disadvantages are that it is very laborious, it's relatively inefficient, you only get heterozygous deletions, and usually it's 1 gene at a time. You can do more genes, but the normal procedure allows you to delete 1 gene at a time. And in principle, it is the manipulation of an ES cell you get from a blastocyst, from a mouse. You manipulate it with very difficult to clone constructs, big constructs, and then you transfect these cells, and you have somewhere you reach the homologous recombination. Then you select with antibiotics the right clone out. You take the clone, put it back in a blastocyst, and then you generate chimeric mice. And then this stage is very important because you hope you have hit the germline. Otherwise, you have to go back and start all over again. Because if it doesn't hit the germline, then it will not be transmitted in the next generation, and you have to start again. If it will be transmitted, you have your knockout mouse. Now, the guys at the Whitehead Institute, Rudi Janisch and his colleagues, they thought when the CRISPR-Cas9 came out, well, maybe we should use that and generate knockout mice with CRISPR-Cas9. And so what they did, use again a similar strategy. They used ES cells, and then instead of using complex constructs, they transfected these cells with Cas9 and the guide RNA. Now, the pathway goes the same, but obviously it's much higher efficiency, and it's much shorter time. You don't have to clone any constructs or anything. It is homozygous, and they have targeted multiple genes at once. So they have targeted 5 genes at the same time, and it worked, and they got mice out of that. So that was pretty impressive. However, they didn't stop then, and they said, well, if we can do it in ES cells, why not do it in the 1-cell stage embryo, which would be even better, because then you circumvent all these problems here, because it doesn't need to go germline, because it is already in the whole mouse once you generate the mouse if you use a 1-cell stage embryo. And that's what they did. So what they basically used was a zygote or the 1-cell stage embryo, and then into this zygote they injected the enzyme, the RNA for Cas9, and their guide RNA targeting any gene of interest. And then what you cause is, of course, a non-homologous end joining or whatever process, or to disrupt your gene. And then you transfer the disrupted oocyte into the foster moms. And then what comes out in the F1 or F0 generation is already manipulated. So we have adapted this as WEHI, or we started to do that as well. And it was probably last year, 1 year ago from now. And then on a Saturday night, I got this email from Andrew saying, "Dear Marco, can we have a holiday on Monday?" Andrew. And I opened the attachment, and what was happened was that, because we injected the BIM guide RNA into mice, and you see we had here 8 different mice born. We couldn't wait long enough, so we took them 1.5 days after they were born, looked in the brain, and the brain contains usually high amounts of BIM gene, as you can see, BIM protein, as you can see here. And then you look through these knockouts and you find, yeah, so you have basically lost the knockout in many of these. In this case, it's 5 out of 8, and then you have 2 remaining, but they have been proven to be mutant as well. So then we sequenced, and indeed we have hit the BIM gene in both cases, and we have interspersed in that, and it worked pretty well, and it was homozygous and everything. And then a lot of people, when I presented, they were saying to me, "Oh, why did you choose BIM? How many BIM mice can WEHI produce? There's so many already there." And then I said, well, it's not so bad, because we know BIM so well, we can really also look for the phenotype. Does it give the same phenotype as a knockout BIM? And this hasn't been done before, and so we started to compare it to the normal BIM knockout mice. So we did this, and we were a bit surprised, because BIM has a feature. If you take away BIM and you take the white blood cell counts from a 6 to 8 week old mouse, then you can see compared to wild type, they're quite increased, so they're hugely increased. Now we look to the exon 3, so we targeted 2 exons in the BIM gene, 1 in exon 3, 1 in exon 2, and you can see that this one reacts exactly like you would expect. But then we targeted the exon 2 of BIM, which also was disrupted, and we couldn't detect it by Western, and it showed normal wild type features. Right, we thought maybe this is just white blood cells, and we went on and looked at another feature of the BIM knockout mice, and this is shown here. The thymus is very, very typical for a BIM knockout. You can see here, this is a wild type thymus where you have the double positive CD4, CD8 double positive cells, and then here these are reduced because you have more single positive and more double negative cells. Now when we looked into the exon 2-3 CRISPR mice, again, the one in exon 2, almost the same as a wild type, one in exon 3, the same as a BIM knockout. Okay, so we went on and did some DES assays, because as I said to you before, in the B cells or T cells, if you treat them with ionomycin, they are resistant if you lose BIM, or if you keep them in culture just. They're usually a bit more resistant to the culture conditions, so we took thymocytes and cultured them in vitro. You can see this is exon 3 and this is exon 2, and this is a BIM knockout, so there it's not as obvious, but where it's very obvious is the ionomycin treatment, again, solely BIM dependent. You look, here's a BIM knockout. Unfortunately, we had only 2 for that. And here's a CRISPR BIM exon 3. You can see it behaves the same, while as the other BIM behaves like a wild type. And here's the same. And PMA is not BIM dependent. We see a little bit of protection, but that's not really BIM. So we were thinking, okay, so what has gone on? So we checked again the protein expression, because obviously the other mice were dead. This was a new batch. And we looked. And what we find is that one of them was wild type, so you would expect that BIM is basically functioning normal. But then the other 4 appeared to us knockout. And then you had the exon 3, which all reacted like BIM knockout, and BIM protein was detectable. So we measured, and we found what we saw was, when we looked at the sequencing, we found obviously number 2 was wild type, which you can see here. It had just 1 base exchange, which didn't lead to anything. And the others had all deletions, which were either in frame or out of frame. But for example, number 3 had this T insertion I talked to you before, which is causing the premature stop codon. And we only found that, and it still did not react like a BIM knockout. And then when you went on and looked for those ones, then we saw that these guys had only point mutations almost, or here 1 deletion and a point mutation on the other allele, but they reacted like the BIM knockout. And so what we found, what happened was that basically the following. So I have to come back to the apoptosis slide. So as I said, the BH3 only proteins they can kill while inhibiting the pro-survivals, and they do this by putting the BH3 domain into, and this is shown here, BCLX for example, they're putting the BH3 domain into the groove, and thereby they inhibit the activity. Now what we did when we generated this exon or these BIM guides was that one of them targeted at the N-terminus, and the other one, the exon 3, in the BH3 domain. So what that means is if you destroy the BH3 domain only in the slightest, BIM activity is completely abolished, and that's what we found. Protein is expressed normally, but the protein is non-functional. Whereas with the one we found at the 5', it seems like even we have destroyed a lot there, the thing is it's not sufficient to inactivate BIM because there must be some downstream start codon or whatsoever, which allows BIM to bind. And another thing was that at the 5', some of them were even in frame, but we couldn't detect it by protein. And the problem was that this is a region for the antibody to bind as well. So the thing I'm showing that is, the take-home message from that is that if you have a new gene you're working with, make sure you're using 2 different guides or more guides which give you similar results. Because otherwise, we know a lot about BIM, we know how it has to react. But if you identify a new gene and you say, well, it does that or does that, just think about it and look carefully what you can do with that. Okay. So now the CRISPR-Cas9 can not only be used for, as I said, non-homologous entering, but you also can induce directed mutations. And you do that basically by providing a template to the cell, and the cell uses this template and repairs the break which Cas9 has induced. Again, the people from Boston, what they've done, they have used it to tag proteins, to introduce reporter into mice, and to also flocks mice. So you can tag proteins, but you also can induce point mutation, and this would allow you to mimic human disease in a mouse model fairly quickly. You could do that before as well, but now you can do it in a much, much faster way. And it was at that time when we were thinking about, right, we want to test whether we can introduce this point mutation, and is it really as easy as it was predicted to be? And at that time, we were contacted by this, and this is a Mission Massimo Foundation. And this foundation is here in Melbourne, and which was founded by a family, and they have a sick son, which is actually him. This is Massimo, and he's affected by genetically inherited disease, which is called leukodystrophy. Leukodystrophy is very, very understudied, and there's not much known. And they were approaching us and asked that because it is supposed that this is a point mutation in one gene, and they asked us if we could make the mouse model for them. And this family is like really amazing what they did. They launched this by themselves. They sequenced the whole genome of Massimo, and they assembled scientists in order to become awareness for that and also to find a cure for leukodystrophy. So again, what is leukodystrophy? As I said, it's genetically inherited. Infants who appear normal, they become sick, and it usually leads to progressive loss of movement, speech, ability to eat, vision, and hearing. And they found the gene, as I said, with the whole genome sequencing. They found a gene which is called DAS gene, and this is the aspartyl tRNA synthetase. So they asked us, could we do this mutation? And we said, yeah, we will help you, and we did that. And so what we had to do was, this is a normal, sequence, and this is in the leukodystrophy patient, so you have only one base pair change, and you get then a protein shift which is from alanine to valine, which is a mutant protein. So what we did, we injected in these mice, we designed guide RNAs around this area, and then we provided the cell with a template oligo which contained the mutant form instead of the wild type form. And so we generated these mice, and I have just the sequencing here up to now. And you can see this is a wild type, and everything which is in green or highlighted in red means mutation. We have some where we get deletions, but you can see there is specifically mouse number 1 has 1 where it's correct, the other allele is destroyed. And then here mouse number 3 has an insertion correctly, and here as well, and here as well. The difference to prior experiments where you had to do that, or where you could do that, is that instead of taking 12 months, we did this in 3 months. So they came in September, October last year, and we had these mice done by February. And they want us to do more because it was not the only mutations. They have now found all over the world, in Washington and in the Netherlands, many children who have that, and it's only focusing around the DAS gene. Okay, so that's one part, to mimic a human disease. But other things you can use it for, the CRISPR system, as I said, is tagging proteins. And we have started to do that as well, and the principle behind that is similar. You have a guide RNA, it binds, it cleaves, and then you provide an oligo, and the oligo has homology, do your cleavage site, around your cleavage site, and then here the red sequence is basically your tag. And so what you end up is that you break that up and you insert this tag into the mice, and this is for a gene we're working on. in the lab, and there's only 5, 6 publications out there, no antibodies, not much known. And so the best way, of course, is you tag this gene, and then you can detect it in vivo. You can do nice Western, or you can follow it by fax, and so on. And the efficiency was really remarkable when we looked into that. This is a DNR PCR for this strep tag, what we used at that time. And you can see out of 28 mice, which we got in the first 2 liters, half of them almost had the gene. Again, had the tag. Again, the speed by which we could do that and the efficiency is unbelievable compared to before. Now, one of the promises as well, which the Enish lab made was, however, that you could use the system to generate flox mice in a similar way by just introducing 2 guide RNAs and then 2 oligos which contain LOXP sites, and then they would be used as a template to be repaired, and thereby you have your flox gene. It sounded quite simple in theory, and we tried the whole thing. So we designed for a gene we're interested in 2 guide RNAs flanking a critical exon, and then we introduced oligos with it. But instead of getting the oligos in there, what we found was, instead of oligos in, we got a deletion of the whole exon. And since we worked in a gene which was important for the fur color, these mice get brown instead of black. You can see they are deleted on both alleles. So we have deleted the critical exon, we have inactivated the gene, but we couldn't get in the LOXP sites. But it was a quite nice thing to see, right, it worked, so we have a deletion. Here the mice are brown, even they're black 6, and here they are brown-black, they're heterozygous. So I have to say that we are now, instead of using the indel formation for making mice, we're using that system for generating mice for anybody, because it's much safer if you delete a whole exon, and then you can work, you know what you're working with, instead of having a mutation in the gene of your interest. And it's easier downstream to genotype your mice by having a deletion of 500 base pairs instead of having always a point mutation or so on. And it's much safer. Nevertheless, we persevered with this gene, and we wanted to have it floxed, because we want to establish the floxing, obviously, as well. And we played a bit around, and instead of using oligos, what we're doing now, we're using circular DNA templates, which we co-inject with the guide RNAs and the Cas9 and these templates, and voila, it worked. We got floxed exon 2. And we looked at this by PCR. This is just a strategy to show that we indeed got that. So this was 3 mice or 4 mice. And you see that this one mouse has been homozygously tagged by this LOXP site. So this is a LOXP targeting. And when we sequenced the LOXP site, it was fully intact. So we had the LOXP site in both alleles completely in. Again, if it works well. And again, the floxing took us then 2 to 3 months. Now that is CRISPR in mice, but you can also use it obviously in this was all black 6 mice, but you can also use it in other mouse strains like the not SCID common gamma, the humanized mice, which are interesting, of course, for many people when you do drug trials and so on. It would be good if you could mutate or convert some of the mouse cytokine receptors or genes into the human genes, because then the background contains a human gene and your cells contain the human genes and so on. And therefore, we thought it might be quite interesting to have mutations in not SCID common gamma mice, but you also can do it in other organisms such as zebrafish toxoplasma in the highlight, or I don't know if it's a highlight, but amazingly last year the first non-human primates were published in Cell by a Chinese group where they made a double knockout monkeys, and it seemed to work extremely well. So as I said, we have done not monkeys, but the not SCID common gamma knockout, and we used P53 just as a proof of concept. Can we now target this strain? It wasn't possible before because there are no ES cells. You can't target it. They're not going germline. So you have to go with something like that, the zygote. You could make transgenics, but nothing else. So we used the system, introduced mutations in P53. We got out of 9 mice, 7 were mutated. So it worked quite efficient. And also the phenotype is tremendous because these mice also, they have no thymus. They got a progenitor tumor, and means they come down after 6 weeks with thymic lymphoma. So this was also quite astonishing. And we have started now 2nd projects in these mice. Just to summarize, this was not all our mice we made. We also had this is what we did since, as I said, more than a year now a bit. So we made basically 6 different indel mutant mice, and this is the efficiency. So you can see this is by far the highest efficiency. When you look, you make a single gene where you have in the NSG. This was also quite efficient. We have also targeted 2 genes already that worked. The efficiency goes down a bit, but it's quite good, about 50%. If we delete exons, what we do now, we have still more than half of the mice which are born are either heterozygous or homozygous deleted in the exon. So very high efficiency. And then if we target set point mutations, about 30% of mice have that. Epitope tagging is quite successful. We have about 50% in these 2 models we made. And the floxing, which is quite difficult, and we are one of the first labs who could do that. We got it to work for one gene. We're working now for some other genes using similar approaches as well. Okay, so then the last bit, and this is just to give you a little bit of flavor, it's not quite excessive, is about the screening. So there were some papers that you could use CRISPR for screening as well. And basically they showed it in 2 human and in 1 mouse cells. And then the idea is quite simple. You have lentiviruses which contain a pooled library, and then you transduce them into a Cas9 target cell. And then what you can do, and we are interested, and especially Sylvia in the lab from Andreas, are interested in necroptosis signaling and which members are involved in there. And so here just an overview of necroptosis. As most of you will be aware, you can induce necroptosis giving the cells stimulating them by TNF and blocking apoptosis and FKB activation. So what she did, she used L929 cells, which you can induce it. She blocked apoptosis by QBD addition and NF-κB activation by IAP inhibition. And then she gets necroptosis. So then she overexpresses the library. And what that means is that most of the cells are killed, but a few will survive. Oops, that was it. Somebody told me I should stop now. Okay, so, and what we basically, okay, and what we basically find was that, so she does this, she stimulates, and then what you get is if you have the library, a few cells will survive because there are some genes, if she takes them away, that will lead to survival. And so that is basically what you see. So first of all, when you look for the guide RNA representation, so the library contains about 90,000 guide RNAs targeting all genes in the mouse genome. So then you treat the cells, obviously you lose the whole representation because the guide RNA representation goes down. You have only a few left, and only this target a few genes. And then we looked which genes did we target, and it was quite impressive because over the last 2 years there were a few papers, Cell Nature, Immunity on necroptosis, and we targeted indeed the important ones, which is the TNFR1, RIP kinase 1, RIP3, and MLKL. And these were our top hits. And the impressive thing compared to shRNAs was that shRNA screens work as well, but the efficiency with what this worked was quite remarkable because you had the TNF receptor, we had 6 replicates, we had 2 independent guides which we found. For RIPK1 we had in 5 replicates 2 guides we found, for RIPK3 again in all 6, and for MLKL as well in all 6. So that means this system is indeed quite, if you have a very strong selection system and a strong bias, this system can be quite useful. And we have of course more hits which are further down, but these were the top 4 really coming up the highest, and we're investigating now what is real and what has to be discarded. Nevertheless, I mean, there's also the advantage, this is a pooled screening, and you can do with pooled screen mainly survival screens, as I showed you today, so you treat everything which survives has an advantage because you knocked something out, so it's easy to identify. But there is other screens where you need an arrayed library where you have basically 1 guide per well, and you have all these 20,000 guides dispersed over 20,000 wells. And these screens include like synthetic lethal screens where you have a drug and a knockout will cause a death. You would lose that guide RNA, and it's quite hard in a pool before and after treatment to find something which you have lost. And the other ones, of course, are like phenotypic screens like migration or morphology of a cell. And of course, if you have the arrayed library, you can do targeted genome subsets. You could say I only look for kinases or the BCL2 family or apoptosis signaling, tumor suppression, and so on. And also the arrayed library will allow you to do individual genes. And we're currently establishing such a library with the SCRF funds we got this year. And the idea would be we get this library as a viral and DNA stock. And so people can do either whole genome screens, or they can pick the arrayed pools by taking the DNA, or they can also do individual clones. And the arrayed is in principle then the same. It will be read out by high throughput machinery up in Liam's division together with Elaine, she said. So then it remains me just to thank the people who did all the work. Again, a big, big thank you to Andrew and Lynn, who did most of the mouse work and a lot of the in vitro and establishing sequencing. They're really stars. And all the others which are in bold here, they have done some work. I've shown some work of the first knockouts in the cell lines. And then here, Gemma, Brandon, they have done the P53 work and the Burkitt lymphoma work. Sylvia and Damien for the necroptosis screen. And of course, Andreas for his long-standing support and mentorship and good discussions and help. Also, Cherry for helping me and very good advice on many things. Philippe for mice and discussions. Daniel, a lot of discussions with him. Catherine as well, who sent out probably 80 MTAs this year for our vector system. She's quite sick of that, I guess. And then we have the systems biology, Liz and Stephen, who did the sequencing from Liam, and then the mouse rooms, of course, everybody, very important and very talented injectors with Fiona and Vicky, who made this all possible for the mouse models. And just to sum up, if somebody is interested in making a mouse, just write at this email, MARCHEC, which stands for Melbourne Advanced Genome Editing Center, and that's what we can do or aim to do. And yeah, just inquire about your model. And that's all.
Got plenty of time for questions. Jerry.
Great talk, Marco. You probably said, but I missed it. When you were trying to knock out MCL1 and you had so many what you call non-responders, what was the explanation for the non-responders? Were that just cells that had lost Cas9?
Yeah, so that was one thing. So the non-responders were originally, they were lower in the GFP and Cherry, which indicates that low guide and low Cas9. But we had also, what was interesting in the responders, also they were highly for these two proteins. We had some which didn't react, and we don't know yet why that is. But the most likely explanation for the non-responders is in the height of Cas9 and guide RNA in the system.
So the first question is, do we find off-target sites? And how do you escalate this? And second, so if in the genome sequence, if there's one nucleotide difference from your guide RNA sequence, would it be targeted by guide RNA?
So to the first question is the off-targets, how we deal with that. So in vivo, for the mouse, we haven't done in that regard too much, but I can tell you that the company I just showed you, they are looking for Taconic. They are looking for, because you get in your programs predicted off-targets, and they're testing with each round of mouse production these off-targets, and they've never found any off-target in the predicted genes. They have found some in some intronic regions, but this hasn't been done too much. The paper I showed you with the monkeys, they have done a whole genome sequencing, and they haven't found any off-targets in these monkeys. In cell lines, it's quite hard. I always say if you grow a cell line, a lymphoma cell line, you would take a genomic isolation on Monday, May the 1st, and on Monday, May 8th, then you would probably get two different mutation status in these cell lines, so it's hard. So how do we circumvent that? What we do usually use 2 to 3 guide RNAs, and if, like with this HRNAs, they give you the same effect, then you can be sure it's not an off-target. The second question was about one base difference in the guide. So when we make the mice, for example, with point mutations, we have to also destroy the PAM site which we used, right? So what that means is we introduce basically one point mutation that wouldn't be sufficient for the guide not to attack again, but as soon as the PAM site, the recognition site for Cas9, which is required for your guide, is gone or has one mutation, it will no longer attack this allele and it will be over. So that means if your guide has somewhere else homology, but with one base difference, you need the NGG in the same distance, otherwise it will be activity of Cas9.
So is it possible to put 2 guide RNA in one cell? Because there is some protein that are more difficult to CRISPR than others. I mean, some genes, sorry. So have you tried, or is there some lab that try to put 2 guide RNA? In fact, 2 guide RNA that like target Exon 1 and Exon 2.
So we haven't done for different genes, for the same gene, but we have done for different genes. So we've knocked out in one cell in Tumor, that's not a problem. So it would be similar what you're saying, that we basically target exon 1 and exon 3 of Tumor, whatever gene you're interested, and thereby you make sure that you can have a knockout either from that, or you get even a deletion and thereby knock your gene out. Yeah, that's possible.
Yes.
What's the largest insertion that you put in that other people would manage to get into the cells?
So the largest insertion, I mean, what we have done was up to now 2 kb, but what others have done, again, back to the company, they have done about, the last one I was told of was about 40 kb. But they had to do it in ES cells in that case, it's quite hard in the oocytes.
Jane.
It's a brilliant talk, Marco. Looking ahead, what do you think, would you like to speculate on the implications for gene therapy? For example, Massimol, taking the Schwann cells and repairing. I mean, it's far-fetched, but if you could just speculate.
It's actually not so far-fetched because there's kind of in the US already there's on the West Coast and the East Coast there's companies coming out of the ground like nothing, like Editas, and then there is like, I don't know who else is working on that, but they all try exactly what you just said. Delivery of Cas. The problem at the moment is to deliver the Cas9 into cells, and it would be an easy one if you have a cell which is mutated, because if your guide would only bind the mutated version and not the wild type, it's quite a safe approach to give it to, and that's what people work on. And I guess it's not so far that we will be there to repair genes. Can you use it for cancer research? I don't know, or for cancer treatments, because you always have to hit everything, so that would be quite hard. But if you can basically give the cell an advantage to grow out, such as in the skip or something like that in these bubble kits, and they can get a fully functional immune system again. This has been done. People have taken ES cells, iPS cells from the bubble kits, have repaired the gene and have put it into an NSG model, and these mice develop an absolute normal immune system again. And there is no trace, because the advantage is, in these systems, you can just do it in transiently, and the thing disappears, and thereby there is no trace in the cell besides this one cut you have induced. So it is doable.
Marius.
So from what I've heard, one of the difficulties to use the Cas9 is quite a big construct. But I think there was some paper recently showing there was a smaller isoform. Have you started working with this smaller Cas9?
Yeah, so we have it now. So we have generated all the clones needed for that. The difference is, of course, you need a different guide RNA as well, because it's different now. The PAM sites will be different, everything. So everything now, which you have all your algorithms, everything is based on the old Cas9. Now you're going to a new one, and I guess it will work in a similar way. I don't see why it shouldn't. I don't think from for exactly that purpose what Jane spoke of, it will be useful because it's smaller and probably easier to deliver for research. But with our system, if it's 5 kb or 3 kb, it doesn't really matter too much anymore. But we use it, yes. And it gives you more flexibility as well. You could target 2 genes differently and so on.
Justin?
So there's a crystal structure for Cas9?
Yeah.
And the small ones?
No. But that's another thing. This group that I showed in the beginning, Jennifer Dudner, they are working to make from the crystal structure Cas9, an artificial Cas9, which just has a requirement that is much, much smaller than the wild type.
Any more questions? Can't see anybody. Oh, yes. Okay, James.
Did your point-muted mouse mimic that but a phenotype, the one that you were using to try and mimic the human condition?
So we have bred them now into homozygosity. So at the moment we don't know, because we need probably some stimulus to induce what's in the boy. We don't know at the moment.
Okay, well, on that note, pretty much.